one of the things that they landed on is based on the analysis was that maybe some of these bacteria don't grow so well on the root. Okay, so if you think about the root, there is a very thin layer of soil that sticks to the root, and all of the life and death struggle that happens in competition and all of that is really happening in that very thin layer of soil that sticks to the root. If you go out away from that root into what is called the bulk soil, there's not that much nutrition, there's not that much water, so there's not as much of that competition happening. That root layer, that layer of soil that it's attached to the root called rhizosphere is where all of this drama is happening. So if you're not able to grow in that layer of soil, there's a problem with being a good biological control agent. Maybe there wasn't enough of the DAPG being produced. That's another possibility. So how do you solve this problem? Do you increase the expression of the genes that produce DAPG? Do you change how that gene set is regulated? Do you put in new genes? Or do you think about other options, for example, to make the bacteria better able to grow on the root? So root colonization clearly is one of the important things. So at this point, we're going to take a slight detour, and I'm going to talk about 3 terms that you may not be familiar with. Number 1 is the word isolate. So if I went out into my backyard and took some soil and shook it up with water and grew it on a bacterial plate, and I collected all the bacteria that grew from there, and I liked one of them because it's a nice orange color, and I decided I wanted to study that, that's an isolate. An isolate doesn't have much biological meaning except for the person who isolated it, right? I picked that bacterial strain colony because it was the right color. Somebody else may pick it because it's good at biological control. So an isolate is a bacterium that you pull out of the soil or from whatever area that you're working on because you're interested in it in some way. It's a little bit like going to the pound and picking up a puppy, right? Now, when you bring that puppy home or you bring your strain into the lab, you have to be able to know which one it is the next time, so you give it a name. When you name an isolate, it's called a strain. So think about naming your dog, it's now a strain. Now, scientists who work in the field tend to have thousands, if not hundreds of thousands of these strains in their freezer. And unfortunately, the naming system for strains is just as varied as there are labs. So I can give you some strain names. They include PF5, PF01, SBW25, Q287, Q8R196, L5196. All of these are strain names. As you can see, there's not much in common among all of these. And that's because each lab names its strains depending on their own set of rules. So the lab that I worked in, Dr. Ramachow's lab, our system of naming it was based on the area that they were identified from and the year that they were isolated. So Q287 was isolated from Quincy, Washington, and it was isolated in 1987. And we'll talk about your strain, L5196, in just a minute. Now you have all these hundreds of thousands of strains sitting in your freezer, and you want to understand how they relate to each other. So up to this point of isolate to strain, you don't really have a biological basis for separating them or grouping them. At this point, you can group them based on an actual genetic analysis, and that allows you to divide them up into groups, and that's what is called a genotype. So a genotype is a grouping of organisms, of strains, based on a specific genetic basis. In this case, what was done was what's called rep-PCR, which is a technique where you identify specific areas of DNA that are common between different strains. And as you see in this picture, you've got different genotypes A through, I think, P. No, there's Q. Okay? And if you look at the A type up above, you can see that they all have a very similar pattern of DNA bands. Okay? And what it turns out is that DAPG producers could be grouped into these different genotypes based on this rep-PCR method. So what does this mean? Why should we care about the genotypes? So it turns out different genotypes behave differently. So the D genotypes are the most dominant in Washington State take all decline soils. Genotypes A, D, and F are cosmopolitans. They're in locations worldwide. And what's very interesting is the A genotype tends to produce, in addition to DAPG, they make two other compounds, pyrrolnitrin and pyrroluteurin. And genotype M is the one that is widely distributed in Western Europe. Okay, so genotypes tend to stay within certain areas. You tend to find the same genotypes in specific areas. So how does this relate to what we're trying to answer? Turns out different DAPG genotypes differ in how good they are at what is called rhizosphere competence. Remember that root with the soil attached to it? Rhizosphere is that soil that is attached to the root, and how well you grow in that soil is rhizosphere competence. And different genotypes were different in their capability to grow in that. So as you can see, you have got Q287, which is the strain that I studied that we identified these genes in. It belongs to the B genotype, and turns out it's not that good at rhizosphere competence. So you can think of Q287 as one end of that capability to grow in the rhizosphere. In this slide, you can see Q8R196, which belongs to the D genotype, is the one that grows best, as you can see from the graph. So different genotypes differ in their ability to grow on the roots, and that's important because we need to understand what is it that makes them different from each other. So root colonization is a complex process, and it's not something that we can say, okay, there's going to be one gene and we'll find the gene and that will be the solution. We expect that it's going to be controlled by many different genes, and that's actually your research project, is identifying those genes that are important for root colonization. So it's fine to see this effect in the greenhouse, but as you can see, that same effect is seen in the field as well. Q287 doesn't do so well. Q8R196 did much better. Another aspect, so I'm going to introduce now the strain that you are going to work on, which is L5196. So L5196 was isolated from a take-all decline field in Lind, Washington. I don't think many of you will know where Lind, Washington is. It's near the gorge for a geographic location for you. And it was isolated in 1996. So if you look at L5196 in its capacity to grow on the rhizosphere, it actually does better than even Q8R196. So you can see that on this slide here, you can see the L5196 growth is much better than even Q8R196. So L5196, therefore, is a key D genotype strain that is able to grow very effectively on the root, and that's why we're sequencing its genome. But L5196 does one other very cool thing, which is shown in this slide. What they did here was they took soil that contained Q8R196, F113, which is another strain that tends to do very well in growing in the rhizosphere, and L5196, and they dried these soils for a year. So it's just sitting around, it is completely dried out, and as you can imagine, that's not very good for the bacteria in the soil when the soil gets dried out. At the end of the year, they said, okay, how well do these bacteria come out of the soil if we start growing them in the lab again? And Q8R196 didn't do very well at all, whereas F113 and L5196 did very well. So they take that sample, they test it. Now they put it again into a dry mode and let it sit for another 6 months drying out, which is again bad for the bacteria. And they do the test again after the 6 months, and then they do it again after another 6 months. And as you can see on the slide, L5196 survives this treatment. So not only does it grow very effectively on the root, but it survives in drying soil under harsh conditions for long periods of time. So we want to know what makes L5196 so good, A, at growing in the root, and B, at surviving in the soil. And that is why you're sequencing the genome of this bacterium. So that's the sort of, that's the research question, what we call the applied research question, that you're trying to answer. Understanding why L5196 is such a good colonizer and is able to survive in the soil is the reason we're sequencing its genome.